Bismillahirrahmanirrahim. The title of our lecture is Autoimmune hemolytic anemia and tolerance, definition, types of induction and mechanisms, and the autoimmunity, which is include induction, mechanisms, diseases, treatment. First, the autoimmune hemolytic anemia, which is type 2 hypersensitivity reaction caused by the autoantibodies against the RBCs membrane proteins, which is lead to lysis of the RBC. And here the RBC, and this is the antigen, which is present on the cell surface of the RBC. Antibody will form autoantibodies against these autoantigens and lead to destruction of the RBC. And the mechanism of the destruction by the type 2 hypersensitivity, which is antigen antibody immune complex, which is lead to activation of the complement and lead to lysis by the classical pathway of the complement, which is the MAP membrane attack complex. And the classification of the autoimmune hemolytic anemia, the warm time, which is either primary and secondary, and the cold type, which is include the primary chronic cold agglutinin disease and the secondary cold agglutinin syndrome, and the paroxysmal cold hemoglobinuria, and the mixed cold and warm antibody type. And the third one and the fourth one, the drug induced like methyl dopant, which is drug used in the treatment of the hypertension. The classification of the antibodies is based on the activity at different temperature and their etiology, as we saw, hot type and the cold type, warm type, and the cold type. The antibodies with high activity at physiological temperature approximately 37 centigrade are termed the warm type, and the cold autoantibodies act best at temperature 0 to 4 centigrade. Patients with cold type autoimmune hemolytic anemia therefore have higher disease activity when the body temperatures fall into a hypothermic state, as in cold weather. Usually the antibody becomes active when it leaves the limbs at which point it opsonizes the RBCs. And when these RBCs return to central region, they are damaged by the complement. Patients may present with one or both types of the autoantibodies. Both are present, the disease term mixed type, because it includes the cold and the warm. The autoimmunity is can be defined as failure of the immunological tolerance. So the definition of the autoimmunity is failure of immunological tolerance. What's tolerance? Tolerance is absence of the immune response in an immune competent person. And this is of two types: natural, which is autotolerance, central and peripheral, and induced, which is called also immune tolerance. The central or the natural, which is divided into central, which is currently thymus, that this can be achieved by the clonal deletion, and peripheral, which is achieved by the T regulatory cells, which is called clonal regulation. And the induced, which is induced by the individual or by a person, which is called immune tolerance, which is occurred in the perinatal period, when the antigen is introduced at perinatal period, and this can be seen by the identical twin. And the adult, which is of two types according to those of the antigen, and here the antigen is called tolerogenes, and they occur in animals, and it's called low zone tolerance by injection of multiple small doses of tolerogene, and high zone tolerance by injection of multiple very high doses of tolerogene. And the mechanism of this is saturation of all receptors. Factors importance in tolerance: the time of induction, nature, dose, persistence, intravenous or oral introduction, absence of the adjuvant, cellular involvement. As we see, T helper cell get tolerance faster than B cells, and T helper cell lost tolerance longer than B cell. Mechanism of tolerance: central tolerance by clonal deletion, which occurs in the thymus, and peripheral tolerance by that the lymphocyte inactivation or is called energy, which is different from the allergy. And here the energy, which is in the lymphocyte inactivation by regulation of the lymphocytes that can recognize self-antigen and peripheral tissue through the Treg cell or T regulatory cell. And the antigen sequestration, or called immunological silence, and here the best example is the eye, cornea, and lens. Immune tolerance in pregnancy. As we know, the fetus, which is considered as the foreign antigen, because half of the antigen is paternal antigen, and also the placenta, which is considered as foreign antigen to the mother. So they may be viewed as unusually successful allograft since they genetically differ from the mother. And in many cases of spontaneous abortion may be described in the same way as maternal transplant rejection. And autoimmunity is the failure of the immunological tolerance. Immune cell will react against antigen causing tissue injury and disease. And the autoimmunity occurs normally as a biological process to regulate immune response. Example, idiotype, anti-idiotype network. And this is the natural process that occurs in the body of the autoimmunity. Causes of the autoimmunity is genetic, environmental, and the immune regulation. When there is a defect in this, lead to autoimmune disease. Autoimmune disease either organ-specific, which is mean that the antibody and the lesion occur at the specific organ. Non-organ specific, that the lesion in one organ and the immunological response described on the another organ. Mechanism of the autoimmune disease: type 2, type 3, type 4 hypersensitivity reaction. Factors that induce autoimmune disease: release of sequestered antigen, for example, I. T helper cell CD4 T cells and its cytokines are more linked to autoimmunity than the T cytotoxic. Modification of the autoantigen by microbes or drugs like the methyl dopa and hemolytic anemia, and the cross-reaction between self-antigen and the microbes like the beta-hemolytic streptococci and rheumatic fever, which is due to similarity between the heart valve and the structure of the streptococci. This leads to cross-reaction between this bacteria and the heart valve. Polyclonal activation, as we see in the Epstein-Barr virus, has lead to polyclonal activation of the B cell. Genetic factor, which is association of HLA-B27 with ankylosing spondylitis. Inappropriate expression of class 2 molecules like thyroiditis, which is an increase in the expression of class 2 molecules, which is increase the immune response. Another factor, hormonal factor, as we know that females are more prone to autoimmune disease due to sex hormone, estrogen hormone. Environmental factor, which is molecular mimicry between cell antigen and environmental particles. The last one, thymic effect in the old age individuals, which is thymic will be involved and decrease in the immune system and increase in the autoimmune disease. Here are some examples of the autoimmune disease caused by goodpasture syndrome, which is type 2 hypersensitivity reaction. This is the mechanism for the immune pathogenesis of this disease, which is caused by the autoantibodies against the renal and lung basement membrane, so it's called syndrome because it's involved in kidney and the lung. Goodpasture syndrome is identified as a trial of glomerulonephritis, pulmonary hemorrhage, and anti-glomerular basement membrane antibodies formation. Patients with this disease may present a spectrum of conditions ranging from pulmonary hemorrhage, renal involvement, and renal failure. And this is the chest X-ray of the bilateral alveolar infiltration in the lower zone. And the immunofluorescence here of the IgG type against the glomerular basement membrane and adjacent tubular basement membrane. Another disease is Myasthenia gravis, which is also a type 2 hypersensitivity reaction, which is a result when the body forms autoantibodies against the acetylcholine receptor on the skeletal muscle, which is lead to relaxation. Here, most significant muscle relaxation is the muscle of the upper eyelid, which is lead to ptosis, which is either bilateral or unilateral. The another disease, autoimmune disease, Hashimoto thyroiditis, which is a chronic lymphocytic infiltration of the thyroid gland, and the immune system will attack the thyroid gland, result in inflammation, and lead to underactive thyroid gland, which is hypothyroidism. It is mixed of type 2 and type 4 hypersensitivity reaction, which is caused by the T helper 1 and T type hypersensitivity and autoantibodies against the thyroid protein and the thyroid cells. And here we found that the infiltration of the lymphocytes, histopathology infiltration of the thyroid cells by the lymphocyte. So during the histopathological examination, we have a gritty sensation when we cut the thyroid gland by the knife. And here the immunofluoresceins against the thyroglobulin and the thyroid cells. The another autoimmune disease, which is the insulin-dependent diabetes mellitus, which is type 2, type 4 hypersensitivity reaction, which is caused by the T cytotoxic cell that infiltrate the pancreatic cell and the T delayed type hypersensitivity cells, and also formation of the autoantibodies against the pancreatic beta cell. And here the immunofluorescein study of the autoantibodies against the pancreatic beta cells, which give fluorescein apple green light. And another disease of the autoimmune disease, which is the non-organ specific autoimmune disease, which includes the first and most important is systemic lupus erythematosus, which is SLE, which is the abbreviation of systemic L for the lupus and the T for the erythematosus, which is type 3 hypersensitivity reaction, which is non-organ specific caused by the autoantibodies against double-strand DNA. This is very important. No single strand is against the double-strand DNA, and autoantibodies against nuclear protein, RBCs, platelets, membrane, and the immune complex are deposited in the kidney. And here we can, it's called systemic because it's affecting multiple organs, the lung, the heart, the kidney, the joints, and also the skin. We found butterfly rash on the skin. The another disease, which is rheumatoid arthritis, which is type 3 hypersensitivity reaction, which is also non-organ specific autoimmune disease. The cause of this disease, the autoantibodies, which is the IgM against the IgG. And autoantibodies leads to immune complex formation that deposit in the synovium of the joints. The genetic factors also contribute in this disease, which is HLA-DR4 and DR1. And here the joints, when we magnify this or make zoom to this area, we can find that the this is the blood vessels and migration of the cells and secrete T helper cells, T helper 17 cells that secrete the interleukin 17, which is lead to the synovial. This act on the synovial fibroblast, and also the stimulate the synovial macrophage, and the synovial T cells, T helper 17 cells will be aggregate, and this lead to secretion of inflammatory cytokines like tumor necrosis factor, interleukin 1, interleukin 6, and this lead to increased rankle, and this lead to stimulation of osteoclasts that destroy the joint and lead to increase the activity of the osteoclast cell that lead to osteoclastogenesis and bone destruction. So we found that the joint will form a soup of the cytokines, and it affects the small joints, interphalangeal joints. And this is the explanation of previous figure. And the autoimmune response in rheumatoid arthritis, the autoantigen is the IgG, which is called the rheumatoid factor, type 2 collagen of the cartilage, and the newest one, the cyclophosphamide protein, is CCP, which is highly specific for the rheumatoid arthritis, and when it is positive, indicate bad prognosis and aggressive and persistent disease. Treatment by the non-specific suppression of the immune system using the corticosteroid in high dose and the chemotherapy cyclophosphamide. And we must concentrate on the immune therapy that only suppress the immune system and not all other highly dividing cells. So we use monoclonal antibodies specific to CD4, T cell receptor, MHC, interleukin 2 tumor cross factor. And as you know previously, this is the diagram show us the stimulation and activation of the T cells, and this is the antigen presenting cells and T cells, and this is the receptors which is involved in the stimulation of the T cells, and we found autoantibodies against these receptors. And peptide blockade of the MHC molecules by synthesis the peptide, which is located in this space instead of the cell antigen. An induction of tolerance by the oral antigen, and which is cheap and easy to administer for application in human disease. And rheumatoid arthritis, one of the autoimmune diseases that had benefit from this form of immunotherapy using mainly collagen type 2. Another transfer of Treg cells can inhibit disease pathogenesis. Another method of treatment, blocking antibody against interleukin 17 and interleukin 23. Statin that reduce cholesterol found that decrease cytokine level pro-inflammatory cytokine and decrease adhesion molecules expression. The last one is generating immune tolerance by using tolerizing DNA vaccine using molecular methods. DNA plasmid encoding cell antigen is translated into dendritic cells, and the presentation of cell antigen by antigen-presented cells without adequate co-stimulation leads to energy or tolerance of T cells because of the lack of interaction between the CD28 with CD80 or CD86, which is the second signal of the T lymphocyte activation. And here, figure show us this is the plasmid and encoding the cell antigen, and this is the dendritic cell, and this is the cell. This dendritic cells will express the cell antigen, and there is no stimulation to the second signal CD28, so there is tolerance. While here the immune activation, this is the non-cell antigen, and this is the dendritic cells which express the non-cell antigen, and this is the first signal of the T lymphocyte activation, and this is the second signal of the T lymphocyte activation, and this is lead to activation of the T cells, which is the co-stimulation. While here in the tolerance, there is no activation of the second signals, so end with tolerance.